Okay, students, here erythrocytic cycles, the pre-erythrocytic cycles, exoerythrocytic cycles we will discuss. Now we are going to learn the erythrocytic cycle. Erythrocytic cycles in the plasmodium vivax in the erythrocytic cycles. The completed in the RBC is called erythrocytic cycles. So it was erythrocytic cycles. It was described by Camillo Galzi. He is the scientist Camillo Galzi. So Camillo Galzi. Erythrocytic cycle. Erythrocytic cycles. It was described by the Camillo Galzi. Hence the name is called Galzi cycle. Hence the name is called Galzi cycle. The 4 marks question important. Erythrocytic cycles. Another name Galzi cycles. Given the examinations, the Galzi cycles. Explain the described by the Galzi cycle or describe the erythrocytic cycles in the plasmodium vivax. So any names in the 2 names in the Galzi cycle or erythrocytic cycles, the both are the same. So because the Galzi cycles is the Galzi cycles, the first of all erythrocytic cycles, it was described by the Camillo Galzi, hence the name is called Galzi cycles. So the first of all erythrocytic cycles, here let us see the clearly observed. This cycle is the pre-erythrocytic cycle. This cycle is the exoerythrocytic cycle. This cycle is the erythrocytic cycle. The 3 cycles completed in man. The first of all erythrocytic cycle. So erythrocytic cycle initiated by. Erythrocytic cycle initiated by. Like first of all, this is the small sizes and big sizes. The small sizes and big sizes. So this is the micro and macro metacryptozoids. This is the micro metacryptozoids enter into the RBC. Enter into the RBC. Exoerythrocytic cycle. Erythrocytic cycle initiated by exoerythrocytic cycles. The completed exoerythrocytic cycles releases the micro and macro metacryptozoids. The micro metacryptozoids enter into the RBC. They enter into the RBC, continuations the erythrocytic cycle. So whenever the micro metacryptozoids enter into the RBC, this is the RBC or erythrocyte. So erythrocytes, hence it is called erythrocytic cycle. So RBC, whenever the enter into the RBC in the micro metacryptozoids, so this is the small sizes, the spiral shape. It transforms into the spiral shape. This is called tropozoites. The tropozoite stage. The tropozoite stage of the micro metacryptozoites. The tropozoite stage of the micro metacryptozoids. The cytoplasm. The cytoplasm push only one side of the push the one sides. So this is the one side of the pushing the one sides, the cytoplasms, the tropozoites, the vacuole is formations. The vacuole, this vacuole is formation. This vacuole is going to do a ring stage. Zimmerson resembles to the finger ring structures. Finger ring like structures. This like structures, hence the name is called signet ring stage. This stage, tropozoite stage transforms. The tropozoite stage, then after vacuole is formation. The vacuoles formations, then after the cytoplasm push only one side the cytoplasm. So one side the cytoplasms, the vacuoles, the finger ring structure is formation. The finger ring like structure is formation, hence the name is called signet ring stage. Signet ring stage, then after. So the vacuoles transforms into the amoeba stage is formation. Amoeba stage, this amoeba stage in the lobophil pseudopodium is formations. Vacuoles transforms into the amoeba like structures. Amoeba is the pseudopodium is formation. The pseudopodiums. The pseudopodium is the locomotory organs capturing the food materials one places to the another places. So moving. So the help of the pseudopodiums. So amoeboid stage, the amoeba have a pseudopodiums. The pseudopodiums that helps. The pseudopodiums that helps the feeds on the cells, the tissues and the host cells, and RBCs, tissues and their blood, the tissues and the debris and the feeds on the host bodies. So this is the cryptozoites and spore plasmodium vivax. So amoeba stage in the feeding stage. Amoeba stages in the feeding stage. So help of the pseudopodium. Help of the pseudopodium, the feeds on the debris and blood, other cells, they feeds on the amoeba stage. So amoeba stage, the feeding, similarly, so this sizes increases. Sizes increases is called stageant. The stageant stage of the plasmodium vivax attains the maximum growth. The growth in the increases, the double. The RBC size in stageant stage, the gradually increases the size, the double. Gradually increases the size in double. The RBC size increases the size in double. This is called hypertrophy. Hypertrophy. Already the previous class we will discuss the hypertrophy. So hypertrophy means cell sizes increases the size. Cell size increases the size. The stageant stage. The stageant stage of the plasmodium vivax, the amoeboid stage, the feeding stage. The feeding stage feeds on the food materials, the host bodies, and next immediately the sizes in the growth and development. The maximum attained growth and size is double, increases the double. So stageant stage increases the size double. So this stage is called hypertrophy. The hypertrophy means increases the size, the cell sizes, the growth and development highly increases the size is called, the volume or size increases is called hypertrophy. The stages of the stageant stage, the hemoglobin. So hemoglobin, this stages, stageant stage or hypertrophy, the hemoglobin. Hemoglobin is the hemoglobin, the soluble hemoglobin, insoluble hemoglobin is formation. The soluble hemoglobin transforms into the insoluble hemoglobins. So insoluble hemoglobins, the insoluble hemoglobins, the granules particles. Granules type of the particles releases the blood. Releases the blood. So releases the blood in the hemoglobins. Hemoglobins type of the granules releases because hemoglobins, the hemoglobin, soluble heme and insoluble hemoglobin granules or dots. So this is the red color dots or red color granules is formations. This granules is called Schepner's dots. Schepner's dots are malaria pigments. So whenever the releases in blood, so hemoglobins, red color dots or red color spots releases. So in the Schepner's dots are malaria pigments, hemoglobins, these spots, this type of the granules, the hemoglobins. So hemoglobin is the toxins. So hemoglobin is toxins whenever the releases into the blood. Whenever the releases into the blood, hemoglobins immediately the toxic natures. So automatically fever. The fevers, the cold fevers and hot fevers, they one by one the fever they causes the malaria fever symptoms appears. So whenever the releases the hemoglobins in blood. So hemoglobins releases and this nucleus several times mitotically divisions. So each one nucleus some number of nuclear bits formations. Each one nuclear bit, the periphery the cytoplasm's number of nucleus is formation. This is called merozoites. So merozoites formations. So merozoites formations and attains the growthing and development. Since the growth and development, so maximum growth and development. So this is the RBC wall. The wall of RBC ruptured. the wall of RBC, the ruptured, the released the merozoites, the released into the merozoites. So released into the merozoites, so merozoites releases into the blood, releases into the blood. So this is the merozoites releases two types, the sizes, the unequal sizes. So unequal sizes, so in the small sizes and big sizes, the small sizes is called male gametes and big sizes is called female gametes. The male and female gametes. So male and female gametes releases into the blood in human beings. So then next male gametes and female gametes releases in the human bodies in the blood. Then next male and female gametes, the next cannot development of the male and female gametes. They can, so this is the male and female gametes, they produces the male gametes and gametocytes, the gametogony, the next continuation of the life cycle. Next another one host, they cannot development any other development in the human beings. Why? Because the blood pH and the temperatures cannot availability of the male and female gametes. So this is the available reaches to the next host in the Anopheles, female Anopheles mosquitoes. The female Anopheles mosquitoes, the female Anopheles mosquitoes reaches to the next one host. So then after the growth and development of the plasmodium vivax. So plasmodium vivax continuations, merozoites releases. So merozoites 12 to 24 merozoites. Sometimes the 12 to 24 merozoites releases in blood. The two types of merozoites, the small and big sizes. Small size of the merozoites is called male gametes. The big size of the merozoites is called female gametes. Male and female gametes they cannot the development in the human blood because the human blood pH and temperatures cannot availability in the, so cannot growth and development, further development cannot in the further development in human blood. So the male and female gametes reaches to the next host in the, so next host in the Anopheles, the female Anopheles. So female Anopheles, whenever the female Anopheles they bite the malaria patients immediately, so they sucking the blood. So sucking the blood through the male and female gametes, they enter into the female Anopheles. So female Anopheles finally reaches to the crop of the male Anopheles. Crop of the Anopheles. So crop of the Anopheles, the oocyst formations, eukaryotes or oocyst formations. Then after finally the sporozoites formations. So life cycle in mosquitoes. The life cycle in the plasmodium vivax, life cycle in plasmodium vivax in man is completed. Then next the life cycle of plasmodium vivax in mosquito. Next life cycles they started initiated by the male and female gametes. So the erythrocytic cycles they start initiated by micrometacryptozoites. Next into the tropozoite is formation. Tropozoites then after the vacuoles is formations, vacuoles one side pushing the signet ring stage. Then next amoeboid stage transforms, amoeboid stage the feeding stage, the feeds in the food materials. Then after the stage stage, the stage stage of the maximum growth in the sizes increases in the double, and it is called hypertrophy. The next stages the hemoglobins, the soluble hemoglobin, insoluble hemoglobin. So hemoglobin is the red colored granules formations, hence the name is called Schepner's dots. The Schepner's dots are malaria pigments. This the hemoglobin is the toxic natures. So hemoglobins in the stages of the nucleus several times in divisions. So each one nuclear bit verify the cytoplasm. So number of nucleus is formation, for example approximately 12 to 24 merozoites formations. So 12 to 24 merozoites and the maximum certain maximum growth they ruptured the RBC walls, they released merozoites. So merozoites releases these cycles, erythrocytic cycles, during this space 48 hours. 48 hours completed in erythrocytic cycle, and 8 days in the completed free erythrocytic cycles. The free patent periods already discussed. Second one is the incubation period. So incubation periods intervals between first entry of plasmodium enter into the blood, formation of sporozoites, and next the formation of the next second entry of the formation of the malaria symptoms. The incubation periods means, what is the incubation periods? The plasmodium vivax enter into the blood, the formation of sporozoites, then next the formation of malaria symptoms is called incubation period. So incubation periods, these periods during the last 10 to 14 days completed. 10 to 14 days completed in the incubation period. So incubation periods, this is the 8 days, 2 days, 10 days, or 10 to 14 days completed incubation period. First plasmodium vivax enter into the blood, formation of sporozoites. The sporozoites, the next second entry into the plasmodium vivax, the formation of cryptozoites is called pre-patent periods. These periods last about 8 days, and these periods, so these periods pre-patent periods, they cannot, so does not appears any symptoms, but incubation periods, the first interval between the first entry of plasmodium vivax, so formation of sporozoites, and the next formation of the malaria symptoms appears. So malaria symptoms appears, is called incubation periods. So this periods the last 10 to 14 days. See the pre-patent periods, incubation period importance, 2 marks questions. What is the pre-patent period and what is the incubation periods? So first this is the important questions. Life cycle of plasmodium vivax man, the plasmodium vivax life cycle in mosquitoes. Any one questions given the examinations, the 8 marks questions. So 8 marks questions given the examination, life cycle of plasmodium vivax. The 3 cycles, right? The 3 cycles: pre-erythrocytic cycle, exoerythrocytic cycle, and erythrocytic cycles. So the 3 cycles in the individuals. Once again 4 marks questions. The pre-patent and incubation periods in the 2 marks questions importance. So this is the, it is clear, this is the plasmodium life cycle in man, and next to plasmodium life cycle in mosquito. The next class I will continue the next sessions. Okay.